All right, here we go. Okay, site-directed mutagenesis. What is site-directed mutagenesis? Does anybody know? If you don't know, rationalize out the individual words. So we should know what site-directed means in the context of DNA. Yeah? No? Okay, so site-directed always in the context of DNA means you're able to like pick an actual specific site. So you're able to pick a sequence within the larger sequence. Mutagenesis, what's that? Making a mutation. So what site-directed mutagenesis is in this lecture, the context that I'm giving it to, typically it's when you have some gene X that you have already cloned into a plasmid, and you might want to make a mutation. So you might want to change some codon, and so you change the DNA sequence while it's inside the plasmid, and now you have a new version of gene X with a mutant. Okay, so that's essentially the gist of site-directed mutagenesis. It's the ability to look at a DNA sequence, pick a codon that you want to change, and then change that codon to whatever you want it to be. That's site-directed mutagenesis. Okay, so as we talk about it today, the context is making mutations on a gene on a plasmid. This is different from site-directed mutagenesis on a chromosome. Okay, so another version of site-directed mutagenesis might be where you have some gene on some chromosome and you want to make some mutation on that chromosome. That is a different process, how you do that, but it usually involves the first step of making the mutation on the plasmid. Okay, so it's just an important understanding to distinguish between making a mutation on a plasmid and making a mutation on a chromosome. Okay. Why would you want to do site-directed mutagenesis? Why would this be useful at all? Any ideas? Yes. Okay. Let me think about this. So it's not going to be the best type of knockout. Remember, we talked about knockouts before, and the best type of knockout of like a gene X would be just completely like removing the gene. So it's not going to be the best type of knockout, but you could get kind of what's called a functional knockout. So a functional knockout would be pretend that gene X encodes some enzyme X. Okay. And maybe enzyme X has a very key cysteine residue. This is just an example. And the cysteine residue on this enzyme X actually causes the activity of whatever enzyme X does. Okay. If you mutate that cysteine to a different amino acid, maybe like alanine, which is essentially an amino acid that does nothing, that's what would produce a functional knockout. So it's still coding for a protein that translates to the same pretty much global structure, but its capacity to do what it does is eliminated. So it's kind of like a functional knockout. Okay, so you could use site-directed mutagenesis to do a functional knockout. Yes. You could also make like a real knockout, like if you wanted to like mutate like the start codon. So yes, okay, I would accept knockouts under some senses. What else can you use this for? Like think of other examples of why you might want to mutate some residue in a protein. Any other ideas? Yeah. So how would I phrase that? I would almost kind of say that as like making controls. So for experiments, but you said it a good way, which is comparing phenotypes. Or perhaps studying natural variation, natural variance. So for example, I've used this example before, maybe gene X codes for the eye color on some organism. And maybe you want to study two different alleles. Maybe one allele has a alanine here, and maybe another allele has a histidine here. And maybe one of these alleles causes one color eye, green eye, and another allele causes a yellow eye. That's an example. So you could use site-directed mutagenesis to create these two alleles and see how the phenotypes change. Yes, that's a good example. While we're talking about that, let's talk about mutation nomenclature real quick. So if you have, let me quick just translate this. Here's our friend Reke from the last lecture. Let me quick just translate this and get the protein sequence. Here's the Reke protein sequence. Okay, so here's a protein sequence. Amino acids, the primary structure, they have a notation. So this residue is called methionine 1, M1. This residue here is K1, 2, 3, 4, 5, 6. Let's do this one. K1, 2, 3, 4, 5, 6, 7. This is K7 here. That's K7. Okay. So this goes on and on and on. And if you make a mutation, you have there's a nomenclature to those mutations. So if I was to say change this methionine to an alanine, so if I was to make a mutation and mutate the protein such that it read like this, the annotation for that is M1A, which means methionine at position 1 has been changed to an alanine. Okay, so we'll do one more. Let's bring this back. Let's do D1, 2, 3, 4. This is D4 right here. And if I change that to K, that's then D4K. Okay. So this is how you annotate mutations. You should know this because I'm teaching you how to make the mutations. So when you make them, you typically have to annotate them. Okay. So it's useful for making controls, making negative controls, potentially functional knockouts, studying natural variations. Another common thing that you use this for is removing restriction sites. Okay, so if this was your gene, here's our Reke. Imagine you had kind of like a nasty internal restriction site. And sometimes this can be a problem where if you have your gene X and your destination plasmid, say plasmid Z, is something very specific that you must utilize. You must clone it into that. Sometimes these plasmids only have a few options. Maybe you only have 2 options for restriction enzymes. So you have 1 and 2, and you have to use these restriction enzymes. You're forced to use them. But if there's a problem where one of these is inside, has an internal restriction site in gene X, you can use site-directed mutagenesis to remove that restriction site by changing the codon to what's called a silent mutation. What's a silent mutation? Yes. Well, it will change the codon, but it won't change the amino acid because there's multiple codons for a given amino acid. So typically what you would do is if there was a restriction site that you need to get rid of, maybe it's this, you can change that codon and just change it to one of the other options so that it maintains the amino acid structure, but you just change the DNA sequence. That's a silent mutation, okay? So you often will utilize this to change internal sequences of genes to facilitate downstream cloning. I've done that like many, many, many times. That's a common thing that comes up. The only other final thing that I would suggest is instead of, it's sort of like opposite of number 1, you can use mutations to enhance protein function. So in 1, you're kind of like, maybe you're taking a protein and you're inactivating it. The flip side to that is maybe you have some protein X, which has maybe like mediocre enzymatic activity. But you know that if you make a mutation right here, it'll give it enhanced or like super enzymatic activity. That's another common thing, okay? So you can use it to change protein function. So these are many things, some of the options that you would use site-directed mutagenesis for. There's many more of these, but these are just a few examples. Okay, so let's walk through the process of like what you actually do when you do this. So here's your plasmid Z. Here's your gene X cloned into the plasmid. So it's pretty simple to understand what you do here. you just design two PCR primers. So one primer will look like this, okay, forward primer. Another primer will have a little bit of overlap with that other primer and go the other way. And in these primers, these PCR primers, at the spot you want to mutate, let's say it's this residue right here, you change the codon. Okay, so you make some change inside of your PCR primers such that when this thing gets amplified, the new construct will be changed at that position, okay? And then you run a special PCR reaction, which the polymerase will come on and it will write all the way around the plasmid in both directions, okay? And then after you've done this, you have produced a new product. So there's a synthesized now plasmid that was synthesized by PCR. And because that synthesized product incorporated those primers, it now has a mutation at the position you chose, okay? In contrast to the template, which still remains the same. That's the gist of what you do, okay? Any questions about that? Yeah. So that's I'll show you what the primers look like. We'll design some. If you bring up an interesting point, it depends on the size of your primers and the size of the difference. So when you do site-directed mutagenesis, typically you're only changing like 1 to 2, at max, 3 codons. I've done a reaction where you change 3 codons, but that's kind of like it. Like so you bring up a point that like if you're going to change a whole bunch of sequence, your primers won't even bind. And so the reaction will get less and less efficient the more you're changing. It works very well for a 1 codon or even a 1 base pair change, right? But it doesn't work so well if you're trying to change a whole bunch of things. There's other methods to change larger structures. So the purpose of this, remember this, I actually ask test questions like this. The purpose of site-directed mutagenesis is to typically change a codon. And that's just enough subtlety that your primers will still bind perfectly fine, especially because these are actually long primers, so like 40 base pair primers. Good question. Okay. So let's go through the steps now. We'll go a little bit more in detail in this process. And then at the very end, I'll walk you through how to design the primers. Okay, so the question is, how does making a new plasmid with a mutation enhance some protein function? So that's going to depend on the protein. So let me give you a more specific example. Let's say you had TN5. Did Kyle talk about TN5, which is a transposon? Kyle, the guy who came in and gave a lecture, did he talk about TN5, which is a transposon? Okay, so transposons are jumping genes. That's their enzymatic activity. They have to excise the DNA, okay? And TN5 codes for some protein TN5. And in all enzymes, so all proteins that do stuff are enzymes. And in all enzymes, there's some amino acid in that protein that is responsible, called the catalytic site, for doing that activity. Does that make sense? And if you change the amino acid at the catalytic site, 2 things can happen, or 3 things. 1, maybe nothing would happen. 2, you break the function, or 3, you make the function more enhanced. Okay, so in TN5, actually what people who use TN5, what they're not using a wild type version of TN5, they're using a version that's been mutated to have enhanced jumping activity. So when you say like, well, what is it about making the mutation that makes the protein have enhanced functionality? That depends on the very specific protein and whatever very specific mutation. Like somebody perhaps studied that and they found that if you change this little position to this, it makes it a better jumper, right? That's very specific. So it's always like a very, very specific thing. My point is that it's very common that you want to change a base for whatever reason. And one of those examples is that you can use it to make a protein more enhanced functionality if you know how to do it. Okay. Another example of it, let me actually just give another example because it's actually a good example. So sometimes, let's say you have enzyme X. A common thing in molecular biology is you'll have a protein and that protein will get phosphorylated by some kinase. And the phosphorylated version of the protein will be more active. So there'll be like these biochemical signaling cascades where some kinase will phosphorylate enzyme X, and then it will all of a sudden start doing its activity. Okay. You can actually make mutations that are called phosphomimetic. So there are certain amino acids that look like they've been phosphorylated. Okay. And if you make those mutations, it can make protein X behave as if it's always phosphorylated. So that's one example. Another example of how you can like enhance some enzyme's function with a mutation. Okay. Okay, before I show you how to design the primers, let's go through the steps quick. So quick steps is one, you're first going to have a mini prep of the plasmid that you're going to modify. So you have to have already cloned a gene into a plasmid, and you've done a mini prep, and you have that plasmid in a tube. Two, you're going to design your primers. I'll show you how to do that in a second. Three, you will do the PCR. So let me run you through this program quick, the PCR thermocycler program, and we'll see how it's just subtly a little bit different. Okay. So in our PCR thermocycler program, first you're going to have your initial denature. This is standard, usually 2 minutes at 98 degrees Celsius. And then you enter your loop typically. So you have your denature, you have your anneal, and you have your extension. Okay, and this does a loop. So denature is usually always the same, 10 seconds, 98 degrees Celsius, depending on the enzyme. Annealing for this site-directed mutagenesis reactions, I usually always anneal at 56, which is kind of like a low annealing temperature. The primers for this are always pretty long, and I've just found by experience that 56 usually like works most of the time. Okay, so you anneal 56 degrees Celsius, 30 seconds. Here's where things start to get a little bit weird. Extension, 72 degrees Celsius, which is normal. But how long should we extend for? How much time should we provide? Yeah. And what's unique about this reaction? It's a whole plasmid. Okay. So if the speed of the polymerase, I think the speed of the high fidelity polymerases are actually 15 seconds per, that's 15 seconds per 1,000 base pairs. So there's some rate at which the polymerase writes, okay? And the unique thing about these reactions is that you're going all the way around the plasmid. And usually plasmids are about like 4,000 base pairs. The backbone is usually that, plus you have your gene X. So you're probably thinking about 4 to 5 kb space that you have to go all the way around, okay? So oftentimes, like you could precisely calculate this out, but usually the best heuristic is usually people will put something like 4 minutes here. So this is one aspect of this reaction that's just a little bit weird, that's different, is you just kind of allow a lot more time in the extension step because the polymerase has to go all the way around plasmid. Okay. The other thing that's a little bit weird is the number of cycles that you do. So usually instead of 35 or 40x, you usually only do about 25x. What do you think is the rationale, or what's a decent rationale for why you have less loops? So because this amplicon that we're producing is so large, what are we very concerned about? Yeah, mutations. Even the best polymerase, if you go 10,000 base pairs, you're going to get a mutation. And so if you did two of these cycles, you're going to have at least one mutation. And then if you do 4 cycles, you're going to have 2 mutations, etc. So when you start to increase the number of loops, you start to accumulate the number of mutations, and it starts to get worse and worse and worse. And so in these site-directed mutagenesis PCR reactions, you tend to have a reduced amount of loops because you're trying to reduce the amount of mutations, but give you just enough synthetic product that you can recover it on the other side. Okay. So these are just two sort of like unique things about this PCR program. Okay. So you run this PCR and it's going to give you a tube. Okay. And in that tube will be all your PCR reagents, plus this new synthetic plasmid, this mutated, and your template, okay? So you have this tube, which is a mixture of the mutant that you made that's synthesized and the old template, which is the old thing that you don't want anymore. So now you have a conundrum of, okay, how do you recover just the mutant one, but not the wild type original template plasmid? So the recovery process involves a transformation step. So you take this tube and you transform it into E. coli to recover it. But before you do that, you have to do something that selectively gets rid of the wild type plasmid, okay? So there's one enzyme a long time ago that I introduced that I said, keep this on the back burner. There's a special restriction enzyme. It was DPN1. Does anybody remember what was unique about DPN1? I said, remember this. It's going to come up later. Okay, so DPN1 is a real funky restriction enzyme that preferentially cleaves methylated DNA. Okay, so here's what you need to remember about DPN1. Usually I probably do put this on the test because actually this is a very important biotechnology reagent. It's a restriction enzyme that cleaves methylated DNA. Okay, so my next question is, of these two plasmids that are in the tube, which one is methylated and why? That's correct. Very good. The original plasmid is methylated. And the reason it's methylated is because it derives from a mini prep. And when you do a mini prep, you set up a culture of bacteria, it grows in a tube, you purify the bacteria, and you purify the plasmid. And so this plasmid is methylated. Why is this plasmid not methylated? Think more at the molecular, that's true, yes, but think more at the molecular level. Like what is it about this one that it didn't get methylated? Why did this one get methylated, but this one didn't? Yes, because it's not in a bacteria or didn't derive from the bacteria, but what was it that's not there in the tube? Well, there's no methylase. So in the bacteria, there's some DNA methylase that's doing the methylation. And when you did the PCR reaction, you did not add that methylase to your PCR reaction, okay? So anything that's synthesized by PCR in a tube by polymerase chain reaction is not methylated. So this is a very important enzyme, and you'll see this recur in these little tricks, these little PCR tricks, because DPN1 is used then as a selection tool where if you add a DPN1 reaction, so the step four is DPN1 treatment overnight. Then overnight, the DPN1 cuts up and destroys this wild type plasmid. And then you have, it won't be completely gone, but you'll have a strong bias of the synthetic construct over the wild type construct now in the tube. So then the fifth step is transformation. Okay. So you take the plasmid, you transform it into, here's E. coli, you transform it into E. coli, and then the sixth step, selection. So you can select on whatever cassette is on the plasmid, and then the seventh step is validation. How would you validate whether or not you successfully produced your mutation? What technique do you need to use? Which one? Yeah, you just use Sanger sequencing, okay? So typically, if you have, here's your gene X, here's your plasmid. Let's say your mutation is right here. Typically, you will have a designed Sanger primer. So remember, Sanger requires one primer. It's like a half PCR. So you'll have a Sanger primer that will read just before your mutation, and then you'll get a sequencing reaction that will span into that mutation, and then you'll get a text sequence that will show you that mutation. And if you get back then your text data, all you need to do is just control find for that mutation. And if it's there, you'll see it there, and you got it. So just one important thing to note is you cannot validate this by restriction enzyme digest or agarose gel, because those are just showing you size. They're not showing you sequence, and the size is not going to change. So this can only be validated by a Sanger sequencing reaction. okay, in general. Okay, so that's the general gist, the general idea. I'll quick do troubleshooting, and then I'll go into designing the primers. Okay, so troubleshooting, things that commonly go wrong. One thing that commonly goes wrong is your plasmid backbone is too large. So I've done probably hundreds of quick change reactions, site-directed mutagenesis reactions. And oftentimes, if you are working with like a yeast plasmid, so remember, yeast plasmids are bigger, up about 6,000 kb. And usually, if I'm trying to do site-directed mutagenesis in a yeast plasmid, I have not been able to get it to work. So this kind of only works on smaller plasmids, smaller bacterial plasmids. Okay, so let's ask, how would you solve this conundrum of you really need to mutate gene X, but gene X is in some yeast plasmid, let's say PS2. It's in a yeast plasmid, and you can't get your quick change reaction to work. What should you do? Yeah, okay, so you're saying, okay, so there's one word for that conveys all the information that you just said. What's the one word? Starts with it's a type of cloning. Subcloning. So it's subcloning. That's the word for what you said is subcloning. You're essentially saying cut out gene X and put it into a smaller plasmid. That's called subcloning. And yes, that's the answer. So subcloning is a common way that you solve this problem. And a while back in the plasmids lecture, I introduced you to this kind of p-bluescript plasmid, which is like a small plasmid, about 3 kb. It's very small. And I think I said something like p-bluescript is kind of important. And some of these other smaller ones are important because they're kind of like building plasmids where you do stuff. You do these reactions on them. And so a good way to troubleshoot this is if your plasmid's too large and you're not efficiently PCRing around the plasmid, you're not getting good quick change reactions, you can just pop this thing out with restriction enzymes. So I'm assuming you had 1 and 2, and hopefully they match in p-bluescript. You can just pop this thing out, subclone it into p-bluescript. And then once it's in p-bluescript, then you can do your quick change reactions, and then you have to subclone it back into the yeast plasmid. So it's a huge pain. It's not fun to do, but in some cases, if it needs to get done, then sometimes that's the only way to do it. So that's one common problem that doesn't always work. Other troubleshooting comes up in the primers. So I'll design the primers next, and I'll show you talk about the pros and cons of different primer setups. Okay, so the gist with designing your primers. The gist with designing your primers is that if you make them look like this, it will work. If you make them look like this, it will not work. Okay, so there's kind of this visual way that you do these. So the length of these is about 40 base pairs. So these are large primers. Okay, and you want kind of the tails of these to overlap just a little bit, and you want your mutation to be right in the middle of this overlap. Okay, and on this side of the overlap, there should be about 4 base pairs. Okay. So the overhang should be only about 4 base pairs. If you do them like this, they just don't work as well, okay? So keep this in mind that visually, this is how you design them. And now I'll show you how to do that at the DNA level. Okay, so here's our favorite gene, Rec A. I had that protein sequence. Where did I put it? Let me quick grab this. Okay, so here's Rec A, and here's Rec A protein. So let's pick a residue to mutate. Let's say we want to mutate this cysteine right here. Okay, let's count where this is. Review word count. So it's cysteine 117. And let's say we want to make cysteine 117 alanine variant. The first question, what do we have to do first? to design our primers, what do we have to do first? Let me unhighlight this. So we picked out our residue. Maybe that's, okay, so primer design steps. 1, pick out residue to mutate. Okay, we did that. What's next? What do I need to do? Take a guess. That's not a sentence. Make it like, okay, so what you want to say is we want to find the codon. Yes. So I've already converted it to nucleotides up here. But the conundrum is, the question is, where is the codon that codes for that cysteine, right? So how do you find that? Okay, so you can take the coding sequence, and there is this program called Expasy Translate. So this is just a online digital translation tool. So you pop in your sequence, and you can hit translate, and it'll give you translations. Okay, and there's different options. So compact is usually if you just want to like look at the protein sequence. But then there's another option that you can click, which is includes nucleotide sequence, which is nice. So if we hit that button, includes nucleotide sequence, now we get the translation of Rec-A with the actual codons. Okay, so now we need to find that cysteine. So the residues is NLLC. So if we search for NLLC, there it is. So that's the cysteine that we want to mutate, and that's its codon, TGC. So now we need to find that. So I'm just going to copy that sequence. I'm going to control find. I'm going to find that sequence. You need to remove the spaces. TGC. Here's the codon. Oh, I did not want to do that. All right, here's the codon we want to mutate. So first thing, now that we found the codon, find the codon. Step 3, remove the sequence for easy manipulation. So I'm going to take this sequence and I'll just pull it out down below so it's easier to manipulate. I'm going to try to grab some on the left of it and some on the right of it. So let's put it down here. Oh, that's not enough. I kind of want that mutation like right in the middle. Let's try this. Okay, this is good. Okay, so I have sort of just like isolated that sequence. This is what I'm going to design my primers from. Okay, so then step 4, all you have to do is change the codon on the computer. Okay, so we'll change the codon on the computer. How do we find the codon that we need? That's an interesting question. How should we pick the codon? Okay, so if we're going to do alanine, there's multiple codons for alanine. What should we choose, and what should be the rationale for why we would choose that particular codon? Any thoughts? True, you would not want to like introduce a restriction enzyme. Yeah, that would be bad. That would be bad. Although restriction enzymes are always, I've never had that like accidentally happen, so I think that's a rare thing. I've never had that happen to me. But you're right, you would not want to have that happen. Well, what other rationale might go into the choice of the codon? That's more okay. So that's more choosing the amino acid. So that is an important decision. Is what should we mutate it to? So if you wanted to keep something conserved, you might choose similar charge. If you wanted to break something, you might choose opposite charge. Or in this case, if we wanted to break the enzyme or whatever it was doing, changing cysteine to an alanine. Cysteine has a sulfur group, alanine has nothing. Like if that cysteine was important, this would definitely like break that site. So you're right that like choosing the right amino acid to mutate to is very important, which is why I ask you guys to memorize the amino acids, okay? But assuming we already decided that, we're going to mutate it to alanine. My question is, there's multiple codons for alanine. How should we choose which codon, which DNA sequence to change it to? Exactly. So that's exactly what I'm looking up. So we talked previously about codon optimization. So the choice of what codon to mutate this to is probably dependent on what organism this is going to be expressed in. I'm assuming this is going to be expressed, excuse me, in E. coli. So let's pull up the E. coli codon usage table. There's one that I like. Is it this one? Here's a decent one. Okay, so here's all the codons for E. coli, and these are the percentages. This looks like bacteria. So let's find the alanines. Where are the alanines? Here's the alanines. It looks like this one is the most abundant one, GCC, at 0.48% compared to the other alanine codons. So I'd choose this one, GCC. Is there any questions about why I made that decision? Okay. GCC. So then step 5. Oh, that was step 4. Change the sequence on the computer. So let's change this now. Let's change it to GCC. So that codon has essentially been mutated now in the computer. And let's make this red just so we see that. Okay, next step. Get the complement. So I'm going to take this sequence. I'm going to kind of visually create a double-stranded sequence of DNA. So I'm going to go to my reverse complement tool. I'm going to paste my sequence. I'm going to change this to complement, hit submit, grab that. I'm going to paste it underneath. Okay, here's a very important thing. When you are working with DNA on Word, you want your font to be what's called Courier New. I'll explain that in a second, assuming I find it. Because Courier New, the spacing of every character is exactly the same. So now you see that the complement perfectly lines up with the other strand. Does that make sense? So this G is binding to this C. This T is binding to that A. This A is binding to that T. If it's any other font, the spacing of the letters is not uniform. That's the characteristic of Courier New. So when you're working with DNA, you want to use this font. Okay. So now we have the complement. Here's the site where we're going to do our mutation. So now we need to actually like decide and design our primers. Okay. So remember when I drew the primers, you want them to look like this pattern, and these overhangs, you want them to be at least 4 base pairs. So in the Word document, what that means is that to the left of this mutation, I need an overhang here of at least 4 base pairs. And I want to start the primer with my GC clamp and end the primer with my GC clamp if I can. So I'm going to start the primer right here, and I'm going to extend up to about 40 base pairs. Let's see what this is at. That's 33. How much is this? 39. I'm debating. Like what I should actually do is I should reduce the size of the font and I should pull more text. Hang on. It's gonna make me redo the whole thing. Hang on. Okay. And this needs to be changed to GCC, which means that this needs to be changed to CGG. This is the mutation site. Okay, let's try this again. Let's see if I can get 40 bases. $37. Okay, so this is the nearest G that makes it close to 40 base pairs. So this is going to be my forward primer. Total size is 38 base pairs. So essentially like the heuristic of the design is that the primers should be as close to 40 base pairs as possible. You don't want more than 40 base pairs because then you start to get charged more money. The longer the primers are, the more money you get charged. So there's kind of this rule on most websites that when you buy these primers, if they're less than or equal to 40 base pairs, you get like a set rate. But you want them long. So that's the rationale for the 40 base pairs. But you also want them to obey the CG clamp rule. Okay. So you're kind of like, it's a multi-objective optimization problem where you're looking for the closest CG and you're looking to bring it close to 40 base pairs. And you're also looking for this 4 base pair overhang. So this would be my forward primer. The forward primer is the exact 5 to 3 prime sequence. The highlighting feature is being very weird. Okay, so here's my forward primer. Let's give it a name. JFB 003 F. It's the forward primer. Okay, and let's annotate these primers. Let's say primers designed to quick change. That's another word for site-directed mutagenesis. Rec A, making the, what was the mutation? C117A mutant. Okay, so there's the forward. Now let's do the reverse. So the reverse on this side, you want 4 base pairs overhanging. Remember to create this little kind of like that little overhang. Okay, so let's start here at this CG. Let's go as far over here as we can, probably to here. Let's see what that is. 39, we can go all the way to 40. Okay, so this is the span of this primer, but this is not my primer, and this is not my primer. Remember, what's the rule for the reverse primer? Say it to me verbally. How do you derive the reverse primer when you're going to actually like order these? So the reverse primer is the reverse complement of the 5 to 3 strand. Okay, and this is the 5 to 3 strand on the top. So what I actually want is I want this, which matches this. I want to take this and I want to take the reverse complement of that. So change this to reverse complement, submit. That's this. Okay, so let's give this one a name, GFB 004 reverse. Okay, so that's it. So just remember this, the rule for the reverse primer. The reverse primer is the reverse complement of the 5 to 3 prime strand. And so to get that, I took the 5 to 3 prime strand and I took the reverse complement. And you can check it. So if it's the complement, that means it follows this sequence, but if it's reversed, so that means this G is this G, and this G is this G. So you can visually confirm that it is the reverse complement. Okay. Okay, that's it. We did it. So like this would work. That would be, you would then just order these and you could do your PCR reaction. Are there any questions about that? So that's how you actually do, it's easy. It's a PCR reaction. It takes 30 minutes to set up the PCR. The DPN1 digests overnight, and then the next day you transform. You can make mutants in 2 days. Very fast. Okay. Other things to talk about quick. We still have 5 minutes left. So there's some situations where you want to make a very specific mutation. That's site-directed mutagenesis. But there are other experimental contexts where you want kind of like random mutations. Okay. So there might be a situation where, let's actually like, okay, so here's an interesting question that you brought up. What was your name again? Skylar. Okay, so you brought up the question of like, well, how might you like optimize a protein? So let's say you had gene X and you wanted to make a version of protein X that had better enzyme activity. but you had no idea what mutation to make. And so you wanted to develop a system where you could test a whole bunch of different mutations simultaneously to figure out which of those might enhance the functionality of protein X. Okay. To do that, you might want to do what's called error-prone PCR. Okay. So error-prone PCR is a way to make random mutations. And what it is exactly what the name states, is it's essentially like a real sloppy PCR. So you have a real crappy polymerase. So maybe you're using like a bad tack. You're using like a bad tack and you're messing with the buffers. You're making the buffers non-optimal so that you're encouraging the polymerase to make mistakes. Okay. And then you would have primers, PCR primers, here and here on your gene. Okay. And then when you run your PCR reaction under mutagenic conditions, when your amplicon gets produced, you're producing a bunch of amplicons that have some mistake. And each mistake is potentially at a different position in that gene X. Okay. And so you can see how if you would do an error-prone PCR, you would produce a tube full of billions or trillions of amplicons that were randomly mutated at random positions. And then you could build a mutation library where you subclone those into plasmids. And then if you had a screening process to figure out like activity of the enzyme, that's one way where you could potentially enhance the activity of an enzyme, even if you didn't know what position to mutate. Okay, so that's error-prone PCR. Let's wrap it up there.